Well, biennial bearing is a major concern in fruit growing worldwide, also in Germany. And we have many susceptible varieties, cultivars, which create productivity losses over the years. So we have that cycling of on and off years, and we estimate that the productivity losses in Germany alone costing the fruit growing industry, the apple industry, around about 35 to 40 million euro on an annual basis. So it's a major concern. We need to understand the underlying mechanism of bearing if we want to solve that issue. And the issue can only be solved by creating new varieties that don't carry this particular trait of bienniality, and existing orchards can only be treated by spray applications of certain metabolites which help to modulate the bienniality of susceptible varieties over the years.
So for a moment, using RNA sequencing, proteomic and metabolic profiling, we were able to identify the first candidate genes, first proteins, and first metabolites that are differentially expressed in buds between on and off trees, prior to floral induction, at the time of floral induction, but also shortly thereafter. Then we started the multi-omics approach, and we were able to detect physiological pathways promoting and inhibiting flower bud initiation. Our results suggest that different metabolites like thiamine or chlorogenic acid and an adenine derivative are involved in early flower bud development. So now we are using this information to establish a model of the genetic mechanisms underlying biennial bearing. For the moment, we have huge data sets, and we have some few ideas how the mechanisms work and how they affect the bearing behavior of a plant. However, we are optimistic that we will have a detailed insight into the regulation of alternate bearing in the next few years.
If there is some tool available that shows the grower when flower initiation takes place, well, then the step to follow is, what can he do to increase the number of buds that are going to be initiated for the coming season? So observing when flower initiation takes place is only one important issue. But then we need to develop tools that start to even promote flower initiation at that point in time to set up a good crop load in the coming year.
We can suggest to apply all those practices which are known to promote flower formation in apple, like annual pruning, irrigation to prevent water stress, for example, regular testing of leaf and soil mineral nutrient content, and the use of fertilizers to maintain optimal nutrient levels to avoid any kind of stress which has an impact on the crop level of the tree. A well-balanced crop load management, fruit thinning, for example, is also suggested to avoid biennial bearing.
Henryk discussed about fruit thinning. There's one point to add. Our research has clearly shown, and this is particularly the work that was done by Dario in Australia, in the Yarra Valley, it is important to thin trees as early as possible. The earlier the tree is thinned, the better the cropping in the coming year. So we need to, we should not, the grower should not delay thinning. It should be done right after flowering or at the point of flowering to take off flowers.
So as we told you, we now have all the data sets available for metabolites, for proteins, but also for genes differentially expressed. And in the first approach, we focused our research on genes which are involved in metabolic pathways. That means we were looking for genes, then we were looking if these genes really resulted in the production of the respective protein, and if this protein was able to synthesize the metabolite. In the next step, we will focus our research on transcription factors. Transcription factors are key genes which are regulating different pathways. And using this information, we will then be able to understand the full process of biennial bearing in apple. And we will use this information to complete our picture and our model. Then we are looking for carbohydrates and also for plant hormones. This is still missing, but this is important because I told you we know that hormones are produced by seeds and are known to inhibit floral initiation in nearby meristems. We know also from literature that carbohydrate competition is an important factor for biennial bearing in apple. That's why we are looking now for carbohydrates differentially expressed in fruit, but also in nearby located meristems. As soon as we have this information, we can validate our model, and we hope to identify key triggers for biennial bearing and then use this information to establish some protocols for fruit growers, which can be applied in the orchard.